Evening and welcome everyone to the BSGCT Public and Patient Engagement Series. The Society have organised these talks to provide a lay introduction and background to the rapidly developing world of gene and cell therapy. We're delighted today to have Professor of Gene Therapy, Simon Waddington, to start this series off for us. Thank you, Simon, for joining us today. Now, I'd like to introduce you to Simon. Simon has been researching gene therapy for more than 20 years. He has worked on gene therapy for muscular dystrophy, cystic fibrosis, haemophilia, and thalassemia. More recently, he and his team have dedicated themselves to developing gene therapies for a range of severe and often fatal genetic diseases which affect young children. Some of these projects are now at the stage where they are planning for clinical trial. He works closely with colleagues at Great Ormond Street Hospital and also collaborates with people in Manchester, Cardiff, South Africa, and Singapore. Today, Simon will be providing us a talk on gene therapy. So over to you, Simon.
Thank you very much indeed. Excuse me. So it sounds like rather a grand title here, but shortly I'll detail what I'm going to be talking about specifically. Firstly, I'd like to thank the BSGCT for allowing me to come and talk to you this evening. As Raj said, I've been working in gene therapy for 25 years, and so I've seen quite a lot of changes during this time. And so I've seen progress from when gene therapy was thought to cure everything, to the point where gene therapy was being written off as a failure, to then its rehabilitation to the point now where it really is starting to see clinical successes. I'd like to first of all thank all the funders who have made our work possible. I won't be talking a great deal about my work this evening, but there are a large number of funders who have been generous in supporting our research over the past 20 years, and you'll see quite a few of those are children's charities. Because we work a lot on gene therapy for early onset childhood diseases. So what I'm going to show you next is the video of someone called Amanda Cardoz. There's a website that's dedicated to her by her parents. She had type 1 spinal muscular atrophy, and so her parents have recorded her life online, so there's a link for this too. Just to warn you, the volume of these videos is a little bit loud, so you might just want to turn down your volumes because I can't control it at this end, bizarrely. And so one is a video of her at around 8 months old, and this, as you'll see, that she doesn't have an awful lot of muscle tone because spinal muscular atrophy. It means that you have very little muscle strength. And then in the second video, you will see her at around 18 months of age. And in terms of her mental development, she's able to think and do these sort of things. But unfortunately, she's got very little muscle strength. So I'll just show you the first video, and then I'll go straight into the second video.
Sonrisa grande. Sì, una sonrisa grande. Che ti viene todo. Besítamela así. Oye.
So, incredibly sadly, Amanda died a few months after that. So she had type 1 spinal muscular atrophy, an inherited genetic disease affecting motor neurons. And it's this sort of disease that really, or one of the diseases that gene therapy really aspired to try to treat. And I know numerous researchers who have attempted to develop gene therapy for this over the years. And it kind of sets a tone for I think for the whole talk that I thought to give. So first of all, unfortunately, I'm going to give you a little bit of a biology lesson. so if you want to switch over to Hollyoaks, this is your time now. And then I'm going to tell you about how our cells work. Then I'm going to tell you how viruses hack our cells, and then how we as gene therapy as a field have exploited viruses in order to deliver genes that we want to deliver for gene therapy. I'm going to then do a series of films where Hollywood has featured gene therapy. And I used to do this as a quiz, but I understand that because this is a Zoom presentation streaming to YouTube, there's a 30-second lag, which means that quizzes don't work. So I'll have to ditch that idea. And once I've shown you these films, after each film, I'll summarize the message that Hollywood tried to convey, and then what we really can learn, or what is useful to learn from these films about gene therapy. And then finally, I'm going to tell you a couple of real-life examples. So now for the biology lesson. Sorry. So it was actually 1966. So this is Edward Tatum, who'd won a Nobel Prize. And he was asked to write a piece about what he thought the future of biology would be, the future of molecular medicine. And he said, finally, it can be anticipated that viruses will be effectively used for man's benefit in theoretical studies, in somatic cell genetics, and possibly in genetic therapy. And this was in 1966. This was before any of the ability to do any of the amazing molecular biology that we did. So it was quite far-reaching to be able to see that this could be possible. So here, first of all, I'm going to show how our cells work, then what happens when we get infected by viruses, and then how we've used viruses to make gene therapy vectors. So this is a picture of a cell, and this is representative of every cell in our body, and probably every cell in every animal, plant, and fungus. It is the same principles. So that big purple spot is the nucleus, and the nucleus contains the DNA. And the DNA is what makes up our genes. So every gene encodes a protein. It's essentially the nucleus contains the cookbook with the recipes for all the proteins in our body. The proteins are our nanomachines that do all they are the structure of our muscles, they are the enzymes in our cells, they are the enzymes that we use to digest our food. They are the proteins in our nerves that transmit electrical impulses and thoughts. So this is how every single cell works. So just zooming in on this. So when I gave this talk the last time, which is probably about 3 years ago, virtually no one outside of biology had ever heard of messenger RNA. Whereas I guess that now nearly everyone in the world has heard of messenger RNA. So now we'll just explain what messenger RNA does in our cells. So we've got a nucleus there, and the nucleus is like the cookbook that contains all the recipes. But then what has to happen is that individual recipes have to be copied out and distributed around the cell for the recipe to be made. And so that copying out, that message that's copied out, is messenger RNA. And then what happens is these little things called ribosomes, they whiz along the messenger RNA and they make the proteins. So here, what you're seeing here is a protein, which is those little pink things dotted around the cell. So now I'm going to show you a cell or a person with a genetic disease. Say, for example, hemophilia. So that would be a mutation in the factor 9 gene. So you can see in the nucleus, the red bit there is a mutation in the factor 9 gene. The message still gets copied out. But here what happens is that these little ribosomes that are turning this into a protein, it can't turn it into a functional protein anymore, so that is no longer made. So just of a list of proteins here that are associated with genetic disease. Hemoglobin is a protein, factor 8 is a protein, the CFTR protein is a protein, dystrophin, survival motor neuron protein, and then a protein called OPN1LW. And these, all when they're mutated, cause these diseases. So when hemoglobin is mutated, it can cause sickle cell anemia or thalassemia. If factor VIII is mutated, it can cause hemophilia A. CFTR mutations cause cystic fibrosis. Dystrophin mutations cause muscular dystrophy. Survival motor neuron protein causes spinal muscular atrophy. And this gene at the bottom causes red-green color blindness. So now I'm going to talk about viruses and how they infect us. So just put aside what I just told you there about genetic mutations, and this is viruses infecting a normal cell. So we've got a normal cell sitting here happily doing its protein manufacturing, unassuming and unaware that it's just about to get infected by a virus. And you've got a virus here in red on the top left-hand corner. And what the virus does is that it attaches to a receptor on the outside of the cell, a receptor that the cell is using, but the virus is hijacked. And now it's delivering its genetic material into the cell itself. So it's hijacked one of the normal cell receptors, and it's managed to make its way and deliver its genetic material. And then what happens is that this genetic material is turned into RNA, and our ribosomes, they will make anything into protein. So here they see an mRNA, not knowing that it's a viral RNA, and they start to make lots and lots of viral proteins. So in other words, the cell has been hacked by the virus. And you get millions of viral particles being produced by the cell. And this general principle is how all viruses work, including coronavirus. So now I want to make a distinction between viruses and gene therapy vectors. So on the left-hand side, you've got a picture of a virus. On the right-hand side, you've got a picture of a vector. On the outside, they look identical. But it's what they carry that makes them different. On the left-hand side, you've got a virus, and it contains genetic material, and it contains the instructions to make the outside of the virus coat. It encodes the message for proteins that deliver the virus to the nucleus of the cell, and it has other encoding that for proteins for escape from the cell. But in gene therapy, what we've done is that we've taken viruses and we've engineered them so that all they contain is the gene that we want to deliver. So viral vectors contain only the gene that we want to deliver and don't contain viral material. So you can't get an infection of a gene therapy vector. It's just doing the good thing for us. So now I want to show a gene therapy vector on the top left. And this is the cell of a patient with a genetic mutation, say hemophilia B. So as you can see, the cell can't make any of that nice pink protein. And we're delivering a gene therapy vector to deliver working copies of that gene. So it acts initially just like a virus. It hijacks one of the receptors. It delivers the genetic material into the nucleus. And then you start to get working copies of RNA out into the cell, and the ribosome starts to make working copies of the protein once again. Now, you can still see little mutant copies there as well. But now you've got the cell able to make protein for the first time. So most gene therapy at the moment works in this way, that you're supplementing working copies to a cell which is deficient because of a genetic mutation. So with that in mind, I now want to show you 4 films and how Hollywood has perceived gene therapy. As I said, normally I would ask people to guess these films, but this isn't going to work in this media. So what I will do is that I will show the film and then I reveal what the film is and then discuss some aspects of the film. So the first film is this.
So we are to have DNA replacement therapy. Let me explain the 2 phases. First, we kill off your bone marrow. Wipe the DNA slate clean. Phase 2, Introduction of new DNA harvested from healthy donors, orphans, runaways, people that won't be missed. I like to think of myself as an artist. And this is when I create. Thank you. It is a painful process, I'm afraid. But all great art is. I'll certainly enjoy working on you.
So the film here was Die Another Day. So on the left-hand side, you've got Colonel Moon, and he apparently gave himself gene therapy to become the person on the right, Gustav Graves. And gene therapy definitely doesn't work that way. So if Hollywood was to be believed, then ex vivo gene therapy is where bone marrow is taken from orphans, strays, and runaways to be injected into the patient and is known as DNA replacement therapy. That is if Hollywood was to have its way. Let's look at what we can learn from this. So the reality is first that genetic therapy is a growing war chest of technologies fighting many different diseases. So there is ex vivo gene therapy, where you take cells out of someone, you do gene therapy on the cells, then you put them back again. You do direct intravenous gene therapy, where you're injecting the gene therapy into the bloodstream, for example, to treat haemophilia. You might do a topical injection of a vector into the retina to treat inherited blindness. You might inject a different type of vector into a tumor to destroy the tumor. Or you might take immune cells out of the patient, reprogram them so that they will recognize a blood cancer, re-inject them, and then they will attack a leukemia. So the point is that there are lots and lots of different gene therapies. Another reality is that most gene therapies to date aren't gene replacement therapy. So as I said before in the little biology lesson, what we've mainly been doing is just adding more working copies of genes. This is how it's worked for most of the time. Now there are refinements to gene therapy, which is called gene editing. And here what we can do is that we can almost cut and paste and edit the genome itself. And that is starting to show some improvements, but it is just part of this whole war chest we have of gene therapy. And then the third point is that ex vivo gene therapy. So what the doctor was talking about there was taking bone marrow from strays and runaways. The whole point of ex vivo gene therapy is that you get your own repaired bone marrow given back to you. So I want to take the example here of gene therapy for X-linked severe combined immune deficiency. So these are also known as bubble boys. So they are boys born without a functioning immune system. They are susceptible to infection, to fatal infection. And so they live inside a sterile bubble awaiting a matched donor to be identified to allow them to have a bone marrow transplant. And what gene therapy was able to do is that they were able to take out bone marrow stem cells from these boys, deliver working copies of the gene, and then put their own bone marrow back again, and then treat them and essentially cure them of that immunological disease. And the advantage of this is that if you get someone else's bone marrow, sometimes the bone marrow can cause what's called a graft versus host rejection, and it can attack your tissues. But if you get your own correct bone marrow back, then it doesn't attack your tissues. So it's been shown now that gene therapy, in some cases, is actually more safe and more effective than having a bone marrow transplant from somebody else. Okay, I'm going to show you the next movie.
The world of medicine has seen its share of miracle cures, from the polio vaccine to heart transplants, but all past achievements may pale in comparison to the work of Dr. Alice Crippen. Thank you so much for joining us this morning.
Not at all.
So, Dr. Crippen, give it to me in a nutshell.
Well, the premise is quite simple. Take something designed by nature and reprogram it to make it work for the body rather than against it.
You're talking about a virus.
Indeed, yes, in this case, the measles virus, which has been engineered at a genetic level to be helpful rather than harmful. And I find the best way to describe it is if you can imagine your body as a highway and you picture the virus as a very fast car being driven by a very bad man, imagine the damage that car could cause. But then if you replace that man with a cop, the picture changes, and that's essentially what we've done.
How many people have you treated so far?
Well, we've had 10,009 clinical trials in humans so far.
And how many are cancer-free?
10,009.
So you have actually cured cancer?
Yes, yes. Yes, we have.
So I don't know if any of you could guess that. You don't get a chance to guess it. But I'm just going to show you a second clip of this film that might help you figure out what it is. Oh, I showed you. On this, actually, I struggled to get the volume down low enough to even make it acceptable. So in the end, I did it without the sound entirely. But as you can see, what's happening here is that Will Smith's character is having a bit of a rough day in the lab. And if Hollywood was to be believed, gene therapy vectors often turn into super virulent viruses, converting everyone in the world into flesh-eating zombies, which isn't the case, probably. No, it isn't the case. So what can we learn from this film, or what should we learn from this film? So firstly, gene therapy vectors are not viruses. As I said earlier, they are missing a lot, if not all, of the viral genes. So therefore, they are, in the mildest case scenario, they are very broken viruses that are missing bits. And now with some of the most commonly used vectors, they contain remnants of the original virus genetic material. So they aren't going to mutate into something scary. The second part is that cancer is another target for gene therapy. So I work on gene therapy for inherited genetic diseases that affect children. And so the gene therapy vectors and the approaches that we use, we want to ensure that, that there's no immune response to the vector, that it is delivered efficiently to all the right cells, and it doesn't cause any inflammation at all. However, this is just one of a wide spectrum of different gene therapies. On probably the extreme end of the spectrum is anti-cancer gene therapy for solid tumors. So here you're delivering gene therapy vectors, possibly to deliver genes that kill cells. So again, completely different from the sort of genes that I want to deliver. I want to deliver working copies of genes, but people that want to kill cancer are delivering what are called suicide genes into the cancers. They also, they want to cause inflammation. They want to create an immune response against the tumor itself. It is gene therapy in exactly the same way, but using a suite of different tools, different but related tools. So again, this just emphasizes that there's a wide range of tools for gene therapy. It isn't just one technology. And a previous member of the British Society of Gene Therapy, Alan Parker, is on Twitter if you want to follow him there. It's Alan L. Parker. And he does amazing work in Cardiff on what are called oncolytic adenoviruses. So I'm now going to show you the third film.
We gave her what we call ALZ-112, a gene therapy that allows the brain to create its own cells in order to repair itself. In biology, this is called neurogenesis. Here at Genesis, we call it the cure to Alzheimer's.
So not sure if you'd guess what that is, but obviously you'll see in the next slide from the fact I give it away, and also the actors, that it's quite obvious what sort of movie it is. I didn't want to spoil it for you. So, if Hollywood is to be believed, gene therapy is passed on to your offspring. It is so potent that one whiff of vector aerosol will transform every cell in your body, and it will make you super intelligent if you're a chimpanzee. Okay, what can we actually learn from this movie? So the reality is that gene therapy is not passed on to your children. It cures the body cells only. So the point of gene therapy is that you're treating only the patient. You're not modifying the human race. That's one critical point. And in fact, it is something that in gene therapy trials, there are rigorous tests to make sure that it is not being transmitted down through the sperm and through the ova. It is forbidden to modify the human race. So gene therapy is only treating your body. It is not treating the forthcoming generations. The second point is it's really hard to deliver genes. And one of the reasons that it's taken us so long to get this working. So apparently in the film, they walk down the cages, fumigating the cages with gene therapy vector to make all of the chimpanzees and other apes intelligent. It has been really, really hard. It is still really, really hard to deliver genetic material into cells. Because as I said before, gene therapy vectors on the outside, in many cases, look like viruses. So our antiviral mechanisms will destroy them before they get anywhere near the cell. We have barriers to prevent their entry, and then we also have specific antiviral mechanisms. And in fact, it has been one of the difficult aspects of gene therapy, for example, cystic fibrosis. Trying to get genes into lungs is difficult enough, but trying to get them into the lungs of patients who have thick, clogging mucus in their lungs is virtually impossible. So it is really hard to get these vectors in, and it's taken us a real long while to develop the technologies to even make it work, even when we are directing it by various different methods directly into the tissues that we want to deliver to. Okay, I'm going to show you the final film.
So let's say you want to change the human body. You want to fix a mistake. You want to repair something, improve something. Well, if you're going to reprogram human genetic material, you need a delivery system, and nothing works better than virus. It's like a suitcase. You pack in genetic mutation, infect the body, and the vector unloads into the target cells. Getting it where you want it, how you want it, is the nightmare, unless you have a map.
So this is the Bourne legacy. I didn't give the film away, but at least I remembered what it was. And last, actually, Hollywood is pretty close to reality. Rachel Weisz's character pretty much hits the nail on the head. Getting the gene where you want it is the nightmare. It has been really difficult to make the technology efficient enough to get enough copies in to make a difference. And it is still one of the areas of gene therapy that is evolving, that we are still developing, is the ability to target exactly the right cells with exactly the right amount of genes. This is still hard to do. We've come a long way, but I think that there is still a lot more work to be done to get it working really well. And the second point that she says is that we're still putting together a viral receptor map. It is true. The tools that we are using, we still don't fully understand the biology. As I'm sure you will have seen from all of the discussions about how the different versions of coronavirus, how they work, how they get into cells, how they spread. And every scientist in the world is working on these. We're still trying to work out exactly how these tools that we use work. And the more we understand, the better we will be in being able to target these tools, make them more efficient, and get them to exactly the cells that we want. So I just want to then show you a little diagram of the progress of gene therapy. And here on the left-hand side, you can see what people think of it and the year. And this is known as the Gartner hype cycle. And it is relevant to pretty much all developing technologies. So I think it was in around 1981 that's what was called the technology trigger. And this was when the first viruses were being stripped out of their genetic material and turned into gene therapy vectors. And it took about a decade for that technology to be good enough to be able to treat the first mouse models of genetic disease. But once those experiments were done in the early 1990s, people started to believe that gene therapy was going to cure everything. So in about 1999, people were convinced gene therapy was going to cure every single disease. There was a huge amount of investment in it. And people were very confident. And unfortunately, there were a few very serious setbacks. So in 1990, 1998, 1999, there was a teenager called Jesse Gelsinger who was injected with an adenovirus vector to treat a disease called ornithine transcarbamylase deficiency. So it was an intravenous injection, and unfortunately, he developed a powerful inflammatory reaction to it, and he died. And it then was revealed that the experiments preceding that clinical trial, not everything had been revealed completely clearly, and not everything was above board. And obviously, there were huge sanctions and prosecutions around this. And around the same time, in a different gene therapy trial, for X-linked severe combined immunodeficiency, the Bubble Boys, 4 or 5 of the patients of the I think 18 that were treated went on to develop leukemia, and it was found that the leukemia was caused by the gene therapy. So this had a huge negative impact on the field. Now, one thing I want to point out about those babies that had ex-skid is that there was no matched donor, so the gene therapy was pretty much their only chance. And of the 5 kids that developed leukemia, 4 of them were treated with leukemia. One unfortunately died. But without the gene therapy, quite likely most of them would not have survived. But nonetheless, it cast a long shadow on gene therapy, and pretty much the field flatlined for probably the next decade. So in around 2005, I went up to speak to the Genetic Alliance. And I said, look, gene therapy, us in gene therapy, we're seeing really interesting results. We think that, it's clear that gene therapy is going to be curing diseases. And the head at the time turned to me and said, oh, gene therapy, we thought it was finished. We didn't think it was working anymore. And this was a really odd situation to be in, because the experiments that we could see that were being published in the British Society of Gene Therapy, was at the time, European Society of Gene Therapy, the American Society of Gene and Cell Therapy. There were incredibly promising results, but it was still a very hard time because the whole field was very much concentrated on ensuring that these technologies were as safe as possible. But from 2008 onwards, I think, things started to pick up because of a few publications, which I'll talk about in a couple of minutes. And I think this brings us to now, where we've now seen quite a lot of successes. We think we've got it working a bit. And there's a lot of encouraging data coming more and more and faster, as I'll just show you. So I think this was probably the index publication of success of gene therapy. And I've spoken about this a few times now already. And it was gene therapy for X-linked severe combined immune deficiency. So these boys, they had the bone marrow taken out, bone marrow stem cells taken out, treated, and then put back in again. And essentially, they grew a new working immune system. So in fact, it was a tremendous success, the first success of gene therapy. It was ex vivo gene therapy. Rather than injecting the vector into them, it was taking the cells out, treating the cells, then putting the cells back again. Unfortunately, this then led to a handful of leukemias. So although it was a tremendous success, it also then suffered from the same concerns over safety. But through the 2000s, there were several really encouraging publications. So in 2008, there were 2 publications on gene therapy for inherited blindness. It's night blindness. So here, by teenage years, boys will struggle to be able to navigate at night, even with street lamps. And there were 2 publications, one from the United States and one from Moorfields Hospital, where they injected a gene therapy vector into the retina of these patients, and these patients were able to see again in dim light. And then I think 2 of the most impressive publications for gene therapy. The one at the top there is in 2011, and these are called CAR T cells. So these were our immune cells taken out of people with leukemia, or taken out of unaffected people. And then these T cells are reprogrammed to attack the leukemia, and then the cells are injected back in again. And they were able to treat leukemias that had been refractory to normal drugs. And in some cases, the leukemias had cleared. So this was a huge area that was just about to go nuclear in terms of the development in treating blood cancers. The other publication down there on the left was gene therapy for haemophilia, and it was gene therapy for haemophilia B. Gene therapy for haemophilia B was always thought of in the gene therapy field as to be the low-hanging fruit. If gene therapy couldn't treat haemophilia B, then it couldn't treat anything. And it took a long time in the coming. I was fortunate to be involved in those very, very, very early experiments there. And the stuff that we saw in the lab told me that this was going to, when it got to the clinic, taken to the clinic by Professor Amit Nathwani, it would treat haemophilia. And in 2011, it did. There were 6 haemophiliacs that received an intravenous injection of an AAV vector, and they all started to make factor IX protein for the first time. And now there are final results coming out. It is likely that these gene therapies will be approved for marketing in the next year or so. Anyway, so this was 2011, and I think at that point, this was a watershed moment. This is where the floodgates burst. And so these are the publications of successful clinical gene therapy that have come out in the past few years. And you can see now, even at the front here, you've got gene therapies here for sickle cell anemia and beta thalassemia, as well as for solid tumors and other inherited genetic diseases. So I think now we are starting to see an explosion in the technologies as people start to get confidence that gene therapy is working in the clinic. Every single gene therapy is a long journey. And for example, some of the gene therapies that I've been involved with, it is a 7 or 8 year journey from conception of the idea to the point of taking it to the first patient. So it is a long journey. So you've got to be in it for the long haul. But I mean, we are seeing complete transformation in these now. It is really encouraging. So I just want now to, and of course, sorry, I forgot the other successful gene therapy, which is the biggest elephant in the room of all, which is genetic vaccination against coronavirus. So there are two types of genetic vaccination. There are adenovirus vaccination and mRNA vaccination, where the RNA is conjugated with lipids. And you've got to bear in mind that these are not new technologies that were just developed 2 or 3 years ago. These are technologies that have been in development for decades. And yet it is showing that the entire world is now being transformed by these amazing genetic therapies. And again, illustrates that this is a suite of technologies. Here you are vaccinating against a disease as opposed to trying to deliver genes stealthily to cure an inherited genetic disease. And I think the future will hold genetic therapies for prevention of Alzheimer's and Parkinson's disease. You'll see immunization against many other diseases. In fact, I suspect that genetic vaccination will become the standard way of vaccinating people. You're looking at clinical trials for protecting against heart disease and stroke, and treatment of lung and liver diseases. So you're looking at diseases here that are acquired as well as infectious diseases, cancers, and inherited genetic diseases. So I just want to close off just by going back to spinal muscular atrophy. So spinal muscular atrophy, as you see, is a horrific disease of motor neurons. And in type 1, this is the most horrific. And just looking at that little red box there, infants that have type 1 spinal muscular atrophy, they will never be able to sit without support. They will have difficulty breathing and swallowing. They can't crawl, they will never walk. And actually 90% of them will unfortunately die before 2 years of age. And so for gene therapy, a group in the States and a group in France, they were looking at delivering working copies of the missing gene, SMN, using what's called an AAV virus. So they did experiments, first of all, in mice. They injected a mouse model of spinal muscular atrophy. And with a single injection in newborn mice, they were able to completely or nearly completely prevent all the symptoms. And the mice, rather than dying at 14 days of age, were living for a year. So then they continued to work. They continued to study the disease. They studied the patients and studied the course of the disease in these patients. And the reason they did this is because they needed to get markers of the disease to be able to work out if their gene therapy, when they applied it to people, whether it would work. So this was their plot of how the disease progresses. So up the side there, you've got what's called the CHOP-INTEND score. So that's how well their motor skills are. So the higher the number, the less affected they are. And then on the bottom there, you've got their age. And as you can see, this massively, steadily declines until, unfortunately, they die. In a cohort of patients, or 15 patients, 15 infants between 1 and 7 months of age, they injected their gene therapy vector. And then they monitored how these children progressed. And I'm now going to overlay this plot against the progress plot of these people with gene therapy. So you can see the line there, the black line shows how they would do without the gene therapy. And then you're seeing how all these colored lines, all of these patients with the gene therapy. And you can see that all of them started to improve on their CHOP-INTEND score. There were 2 at the left, at the very top, that were injected when they were youngest, and they've exceeded a score of 60, which means they are indistinguishable from unaffected children. So this is absolutely transformative for the families of infants diagnosed with spinal muscular atrophy, And this now is a licensed gene therapy. It's also licensed on the NHS. It's called Zolgensma. And there are patients treated now, I believe, at Great Ormond Street. And I think also now the first ones are being treated in Manchester. And I just want to finish off with a video here. This is Evelyn, who was one of this cohort that was injected with the gene therapy vector.
Now 3-year-old Evelyn challenges Dr. Mendel to a dance-off at her annual follow-up appointment.
She comes back after 3 years and she runs up to me and hugs me and says, Dr. Mendel, I love you.
I forget that she has SMA. This is a healthy girl and she does everything that a normal 3-year-old child would do.
Something like that has never been achieved before.
With our first daughter, it was just devastating to lose a child. You lose all dreams you had for their future. And now we can actually save for college.
So, in conclusion, I've told you a bit about how our cells work, how viruses hack us, how we've hacked the viruses to make gene therapy vectors, what Hollywood makes of it all, and real life. So I'd like to thank you for your attention, and I'll be happy to take any questions. Thank you.
Thank you very much, Simon, for that great talk. So we do have some questions for you. The first question is, when we supplement functional copies of the gene, what prevents the gene fragment from integration into the human genome? And also, what ensures it functioning to produce supplemental copies?
Okay, so I guess the answer to the first question is that, so some gene therapy vectors, they are designed not to integrate. They remain outside of our DNA. And so they work fine for a long time. And this is how the kids with spinal muscular atrophy, they get a vector called AAV. It doesn't integrate into the genome. And as you can see, is that because it's going to nerve cells, it lasts for a long time because we don't turn over our nerve cells. We don't get new nerve cells. Whereas in other gene therapies, like the X-linked severe combined immune deficiency, you're taking out stem cells and you're putting in a supplementary copy. And this is just a small number of stem cells that has got to grow into an entire immune system. So in this case, you've got to have the gene integrating into the DNA of those stem cells. Because if it didn't integrate, then it wouldn't work. It would just get diluted and you'd lose it. So for some vectors, you don't need it to integrate. For some diseases, you have to have it integrating. What was the second bit of that question?
So the second bit was, what ensures it functioning to produce supplemental copies?
It is like a little mini version of the whole gene. So it has control elements that switch it on. So the nucleus thinks it's a gene, even though it may be floating around separate from our DNA.
Great, thank you very much. The next question is, what are your top tips for ethics committees to look out for in gene therapy studies?
Wow, that's a tough one. I mean, obviously, ethics is a huge part of being able to translate this to the clinic. So, one thing that's critical is that the people that are developing the gene therapy can't be the ones that are advising the patients to run the trial, because obviously is that you want people to be trying your gene therapy, but so you need someone independent who is able to give independent advice to those patients, so the patients truly understand the pros and cons. I think another part of this ethics is for the ethics committee to understand the benefits themselves, the benefits and the risks. So, for example, with haemophilia was quite difficult because haemophiliacs, they already had injectable clotting factors that treated their disease. Now, it didn't cure them, and they would still get bleeds, and they would start to get joint damage, but it managed the condition. Gene therapy promised that it would mean they would never have to inject themselves every week, and they would never get joint bleeds. So the whole thing is a risk-benefit analysis that has to be made for each therapy, I think.
Great,
so
thank you very much.
It's conflict, I guess. It's making sure that people don't have conflict and self-motivation, and also then making sure that the patients are generally going to benefit from this.
Great, thank you, Simon. And we've got one last question for you. Thanks, Simon. You focus a lot on viral gene therapy. What is your assessment of how non-viral gene therapy stands within the field?
Okay, so even before the mRNA vaccination with coronavirus, I was starting to get the impression that viral gene therapy is kind of like just kind of the foothill of the way that gene therapy is going to progress. Because viral gene therapy, it delivers very efficiently working copies, but then you can't take them back out again. Once you put them in, they tend not to go away. Whereas really genetic therapy is going to be used for treating diseases where you want to have something expressing for a month, 2 months, and then you want it to go away. So I really think that the future of gene therapy is going to be non-viral. I think where you are able to give people a dose of non-viral therapy that will last for a few weeks, a few months, and then it will go away again. And we've seen how effective non-viral gene therapy has been with RNA vaccination of BioNTech, Pfizer, and Moderna. So I think here we're going to start to see an explosion in non-viral delivery methods. Yeah.
Great, thank you, Simon. That's all the questions we have today. I'd just like to thank you on behalf of BSGCT for starting off this series for us. So thank you so much. Thank you for the great talk. I'd just like to end this session with just highlighting the 3 different talks that we will have coming up for BSGCT. So Simon's going to share a screen with us.
Oh, sorry.
Thanks, Simon.
Yeah.
So the next series that we've got running for BSGCT are research seminars in February, and we've got the ECR seminar in March, and we'll be hosting the next talk for the patient and public series in April. So thank you so much for everyone joining us today, and hopefully we'll see you guys soon in April. Thanks.
Thank you.